Hi everyone. So in our previous video, we talked about the TNF and FAS signaling pathways. For this video, we're going to talk about kind of intrinsic apoptotic signaling and some of the proteins that are specifically involved in the initiation of apoptosis, right? So a lot of apoptosis kind of revolves around, if I start to draw it out here, maybe I can get a nice image. There we go. It revolves around the mitochondria, which looks something like this. Oh, let me turn off that feature. Here we go. Around our mitochondria. This is my mitochondria. And it's going to involve the release of various pro-apoptotic factors from the mitochondria. So there's a couple different proteins that can kind of play a role in this process. I'm going to put up our first type, which is our anti-apoptotic proteins. I'll call them anti-apop proteins. You might also see these called pro-life proteins. And these are proteins that are present on the membrane of the mitochondria, the outer membrane, that prevent apoptosis from happening. So our two big examples are BCL2. Let me write that a little bit better. BCL2 and BCLXL. Those are two examples of our anti-apoptotic proteins. And in particular, what they do is they prevent the formation of pores on the outer membrane of the mitochondria. Pores on the mitochondrial outer membrane. We'll just call that MOM. Right? So if we can't form pores on the mitochondrial outer membrane, then we can't get this release of pro-apoptotic factors. So that's the role of our pro-life proteins. We're also going to have our pro-apoptotic proteins. And our big examples that are directly interacting with the mitochondrial outer membrane are going to be BAC and BAX. And these two proteins are responsible for interacting either with themselves or with each other in pairs to form pores. So what that might look like, I'll go to another portion of the membrane, maybe over here, is I would have two of these proteins come together. Maybe this one is BAC and this one is BAX. And this would lead to the formation of a pore right here in the mitochondrial membrane that would allow for the release of some pro-apoptotic factors, which we'll talk more about in a few minutes. So that's really the main role of our pro-apoptotic proteins, and kind of our inhibition would look like this. If we would have something like BCL2 that's present, and it would interact with a protein like back, and it would be kind of inhibiting back's ability to form pores, right? So that's what our inhibitory activity would look like. Let me maybe just label that. So this is our inhibitory activity. So this is preventing apoptosis. And then this right here would be our pro-apoptosis activity. So progressing apoptosis, right? Okay. Then we do have a third type of protein, which is called a death helper. I'll draw that right here. Death helper. The three examples are BID, BIM, and BAD. And these have two main activities. The first is that they inhibit BCL2 and BCL-XL. We'll show how they do that. And then the second is an activity called KISS and run. So let's go back down to another portion of the membrane to demonstrate what those two activities might look like. So if we go right here, we have one of our pro-life proteins. Maybe this is BCL-XL here. Our death helpers, maybe this is BID, are going to come and directly inhibit BCL-XL, and they are going to prevent interaction with our pro-apoptotic proteins back and backs, right? So we're preventing interaction with those proteins. The second thing that BID and our other death helpers are able to do is actually directly deliver our our pro-apoptotic proteins to the membrane. So if I have a death helper like BID and I have backs right here, it can come in and bind to backs. So if I redraw this in this next step, it's now bound to the backs. So this is step 1, this is step 2. And then lastly, it will insert backs into the membrane and then unbind to leave. Right, so this is still our Bax molecule. Right, so it comes in and it binds to Bax. It inserts it into the mitochondrial membrane, and then BID goes off to continue doing whatever actions, either more kiss and run, or it could be engaging in inhibition of those pro-life proteins. So again, this is a representation of that kiss and run activity. So those are our two roles of our death helpers, right? And we will talk about kind of the process of what's called the MOMP pathway and the release of these pro-apoptotic factors from the mitochondria in a second video. So we will return to this topic in the next video.